From what we have discussed in the previous segment, it is now clear to us that there exists a relationship between oxygen-free radicals, reactive oxygen species, cellular damage, and aging. And uh, based on the couple of research articles that we also discussed in the previous segment, we already are familiar with the idea that there is an inverse correlation between lifespan of individuals of a particular species and the rate of oxygen consumption. Or in other words, an inverse correlation between longevity and the rate of metabolism in these individuals. Now, the idea about this relationship has its origin in the work by Max Rubner, who was best known for his work on energy metabolism. And he was interested in looking at the relationship between the rate of metabolism, or in other words, the rate of oxygen consumption, and the body mass. Now, it was he who in the first decade of the 20th century basically was the first one to propose or discuss that there is an inverse correlation between the rate of metabolism and longevity in eutherian mammals. And later on, in 1928, the idea was extended further by Raymond Pearl when he proposed what is now referred to as the rate of living hypothesis. Now, what is this hypothesis? This hypothesis says that, look, the amount of energy that can be spent or the amount of energy that can be expended in a given lifetime is limited. So automatically what it means is that if you spend the energy faster, that means if your rate of metabolism is faster, then lesser will be your lifespan. This is just another way of stating the inverse correlationship between the rate of metabolism and the lifespan. Now, at that point of time, this hypothesis had its share of support from whatever was known at that point of time. Say, for example, if animals are exposed to conditions which have very high oxygen tension, much larger than what is there in our atmosphere, then that leads to toxicity and therefore aging. On the other hand, in around 1930s, it was realized that there seems to be a relationship between hyperoxic death and metabolic rate. They are interrelated. For instance, in hyperthyroidism, which leads to high metabolic rate, you see accelerated hyperoxic death. On the other hand, hypothyroidism, which lowers the metabolic rate, causes a delay in hyperoxic death. Fine. So at that point of time, the free radical theory of aging was just in its infancy. Now today, the rate of living hypothesis is not accepted because of its shortcomings. But what it did, of course, is to draw the attention of the researchers to oxygen and oxygen consuming metabolic processes, and that paved the way for the free radical theory of aging. Now, free radical theory of aging had its own share of problems in the beginning, because even until the middle of the 20th century, it was believed that yes, oxygen free radicals and reactive oxygen species do exist, but then they are so reactive that their lifetime in the cellular milieu is so small that they cannot really cause any damage to the components of the cell, be it proteins, lipids, or even DNA. But later on, it was realized that under the influences of several factors, for example, oxygen poisoning or maybe radiation. So under these influences, free radical oxygen or oxygen free radicals can be generated endogenously, and they can cause severe damage to the cells, and they can lead to toxicity. So in the year 1956, Denham Harman took a cue from the rate of living hypothesis on one hand and the understanding that radiation can cause mutations, which in turn can cause cancer and aging, and proposed what is the basic free radical theory of aging, in which he said that oxygen free radicals generated by oxygen metabolizing processes do cause oxidative damage, and they are essential for the aging process. Okay. But then again, the free radical theory of aging was dealt with skepticism, because people were not still sure about the existence of free radicals even until the middle of the 20th century. Later on, some developments did happen. For example, investigators detected hydrogen peroxide or oxygen free radicals in cellular environments. Then there was discovery of superoxide dismutase, and gradually the free radical theory of aging began to be accepted. And it was in 1972 when Denham Harman modified his earlier free radical theory of aging, and the modification was such that he introduced mitochondria into the picture. This was basically in recognition of the fact that mitochondria is a seat of production of reactive oxygen species, or oxygen free radicals, and that we now know is because of the oxygen consuming processes that happen in mitochondria by way of performing the oxidative phosphorylation and the electron transfer chain. Okay. Now after Harman's modification of the theory, later on people also brought in other factors which are technically are not free radicals. Say for example, let's say peroxides. Okay, they are technically not free radicals, but nonetheless they have the ability to cause oxidative damage. So post Harman's modification of the free radical theory, what came into being is the oxidative stress theory of aging, which encompasses all other compounds which are present in the cellular environment like peroxides and aldehydes, which have the property to cause oxidative damage. And the refinement of the earlier theory into the oxidative stress theory of aging also took into account that under normal conditions, when the cell physiology is absolutely normal, even at that point of time there is continuous production of this oxidation causing molecules, the reactive oxygen species or these free radicals. And During the process of aging, the oxidative damage increases gradually, and that increment in the oxidative damage which gets accumulated in form of damaged proteins or damaged nucleotides in the DNA, they ultimately lead to aging. Okay, so uh, in the years following Herman's proposition of the modified free radical theory of aging, which also included mitochondria, several experiments were done to find support to the basic aspects of Herman's theory. The first aspect being that oxidative stress increases with age. In fact, the couple of articles from the Bruce Amisis group that we discussed in the previous section or the previous segment are in fact studies which offer supportive evidences to the fact that as the age increases, you do see an increment in oxidative damage, and that increased oxidative damage is basically associated with the process of aging. But what about the other aspect of Herman's modified free radical theory of aging? That is the involvement of mitochondria. And that's what we are going to deal with in the next part in this particular segment.